Hello everyone. Structure rearrangements like inversions, translocations, and duplications are a major source of genomic variation. However, it is still challenging to identify them accurately, partially because current methods try to identify them using sequencing leads. Here we present SyRI, which identifies all classes of structure rearrangements using whole-genome assemblies. In addition to structure rearrangements, SyRI also identifies SNPs and indels in all syntenic and rearranged regions. This provides a hierarchy of variation, which would be helpful in improving the performance of GWAS and recombination analysis. SyRI outperforms current methods in simulated as well as real dataset. We've experimentally validated more than 100 of the predicted translocations using a genetic approach. SyRI is an open source method and can be downloaded freely from GitHub. For more information, please check out our poster and our paper.